Today's daily video is a review. And so I'm not going to spend a lot of time on it. If you have questions, please be sure to contact me in any way. Also, we are having 2 Google Meets a day. So if you want to come and ask questions, that's where you should be coming. So this is just a quick review. Homologous chromosomes are the ones that code for the same genes. So they have the same genes on them. And so like this is the one from a paternal chromosome, and that's a maternal chromosome. They both have a gene on them for hair color, like right here. And so they then code for the same thing. They may be a different allele. So this may be for brown hair and that may be for like red hair, but they are still coding for the same thing, hair color. So we call those homologous chromosomes. They're the ones that are numbered. So this is number 1 from mom, number 1 from dad. Okay. When they go through DNA replication, they form this beautiful X shape. Those are called still homologous chromosomes. The green is one, the purple is the other. Sister chromatids are, of course, the identical copy that was replicated in DNA replication. Okay. Tetrad, this is a fuzzy picture, sorry. I missed this for some of you guys in class. We didn't get to it. But a tetrad is basically something that is formed only in meiosis. You need to know that. Only in meiosis, where chromosome 1 from dad, chromosome 1 from mom physically join up. And they do this in meiosis, and then that way, whenever you create 4 little haploid cells, you would have like one sister chromatid here. That would go to one of the 4. One sister chromatid here. That would go to another 4. One there, one there. You get the picture. Each of the haploid gametes, the sperm or egg, get one of these sister chromatids, and that way they get all the genetic information. It's just only either the genetic information from mom or dad, or from, that lineage. And so they don't get like one copy of the maternal and one copy of the paternal. They just get one copy total. And so in order for that to stay together, to make sure that each one goes, each cell gets one of these, they actually physically attach to each other during metaphase of mitosis. Of meiosis. And that way, whenever they split, the cell is ensured that it's getting one of each. Okay. Haploid means you only get one of each chromosomes. That would be a sex cell, a gamete, sperm or egg. Diploid means you're getting 2 copies, one from mom and one from dad. Most of your cells are diploid. That would be all of your body cells, anything that's just doing normal work. Your sex cells, be it sperm or egg, are haploid. Okay, and then 2 haploid cells join together, fertilize to make a diploid. Okay, this is a diagram you absolutely need to be 100% familiar with. You have your 2 end cells in both of them. You have your diploid, that's called a diploid cell, in both of them. It goes through DNA replication where it makes a copy, even though you have sister chromatids now, and so you have double the amount of DNA. You'd still only have 2 ends. So we don't count those as their own chromosomes. They are identical to each other. So there's no new or different genetic information there. So now exactly the same to this point. And then all of a sudden there's mitosis, just brings it right back down to what the mother cell was. These are identical to the mother cell. Therefore, replacement growth, asexual reproduction, stuff like that. These are somatic cells. That is another word for like body cell. Like they're just a normal cell. Not so in meiosis. Meiosis 1 already splits them to haploid. Why is that? Because whenever you're looking at a tetrad, the dad's sister chromatids stay together. Weird. Mom's sister chromatids stay together. So this cell only has like a paternal chromosome number 1, maybe a maternal chromosome number 2, maybe a paternal chromosome number 3, maybe a maternal chromosome number 4. You get the picture. It doesn't matter if it's mom or dad, if it's the chromosome came down from maternal line or paternal line. It's just the fact that it only has either one or the other. It does not have both maternal and paternal. The sister chromatids stayed together. The homologous chromosomes broke apart from the tetrad, okay? And so we're already haploid. We already only have half the genetic information. We just need to divide it out so we don't have too much DNA in each cell. Now we're down to only 23 chromosomes in each one for humans. Of course, if you're talking a different organism, they would have not 23, it would be some other number, okay? Humans have 23 pairs of chromosomes. Other organisms have different numbers. Okay, these are gametes. Gametes is another word for sperm or egg. Make sure you're writing all this down if you don't have it already. Spermatogenesis, I just wanted to show you the difference. This is if you're making sperm, you start with one big 2N cell. You're still 2N after replication. You go that, split them up, meiosis. You're already haploid. You've got your little 1N there. then you have 4 cells, which then mature into sperm cells. Okay, difference is in oocytes. This is a terrible picture, sorry. It's very grainy. In oocytes, this is eggs, it's making an egg. Just love that term. Looks like oocyte, but it's oo. So in oocytes, look, you're splitting into one big and then one polar body. And then the polar body split. And then one big, one polar body. So instead of making 4 different eggs, like you did in the spermatocytes, you're only making 1 egg. And the rest of them are just polar bodies that are reabsorbed into the female body. And this is the one that is released to see if it's fertilized. Okay, so here is a picture of meiosis. So you're ending up with 4 haploid cells. And then of the 4, one of them perhaps may fertilize an egg, or I guess, I mean, it looks like sperm to me because you don't have 3 polar bodies in one huge egg. And so this would, if this was humans, you'd have to pretend like there were 23 chromosomes in here. So maybe there's 22 hiding behind there. So you have your sperm, it fertilizes egg, and voila, you have a zygote. Oh, you can barely see that word. Zygote. A zygote is a fertilized egg that is now diploid. So the whole purpose of creating these haploid cells is they can meet up with another haploid cell and make a diploid cell. That's a zygote, which will be an organism someday if all goes well. And this is where a karyotype comes from. So a karyotype looks like this. This is like all the chromosomes for one human individual spread out onto a little piece of paper so you can see what the heck is going on. There you can see the banding patterns in them. So this is like chromosome 1, and that's chromosome 2. So 1 from mom, 1 from dad. So you have 2 chromosomes there, but they're only chromosome 1, just a homologous pair of chromosome 1. And then here's chromosome 2 and the homologous pair of them. So you go all the way down, get smaller and smaller and smaller and smaller and smaller until, woo, this is a weird one. So this is particularly strange because this one, you have a long one and a short one. This is a non-homologous pair. This is the sex chromosomes right here. Chromosome 23 identifies your sex chromosomes. Males, boys, whatever you want to call them, have the non-homologous pair. They have an X and a Y. And so they actually don't code for the same traits. They don't have the same genes on them. If this was a female, they would have 2 long ones. They would have 2 X's. Write it down. Females are XX, males are XY. Females have 23 homologous pairs of chromosomes. Males have 22 homologous pairs and 1 non-homologous pair. Autosomes, this is your vocab over here. Autosomes are these guys. They all are just like the same for males and females. And then sex chromosomes are that last pair. Okay. The only thing you check with karyotypes is you scan through them and you're like, do they have 2 of each? Because if you have 3 of each, there's one right here. This one only has 2, but you could have 3 right there, and that would be Down syndrome. Chromosome 21, a trisomy, tri meaning 3, some referring to chromosomes, trisomy. 21 is actually survivable. There's also Edwards syndrome, which is trisomy 18, and there's Pateau syndrome, which is trisomy 13, and there's Klinefelter's, which is trisomy 23. But people can be Klinefelters and you can hardly even notice. They just have an extra X chromosome. So they express male. Pateau syndrome and Edwards syndrome, those trisomy 13 and trisomy 18 are usually fatal at a young age. And the rest of them, they won't even be born. Like if you had a trisomy 1, it would just be spontaneously aborted. You'd have a miscarriage on that one because it can't survive that, or 2 or 3 or 4, any of them. Okay. So I just wanted to get down to the genetics of chromosomes. So when we're doing Punnett squares, this is what I wanted you to understand. So here's our 2 homologous chromosomes, 1 from mom, 1 from dad. Actually, I did that backwards. Look up here. It tells me paternal is purple, maternal is red. And basically, so here's a gene. Let's just say here's a gene for, let's say cystic fibrosis. Ooh, let's not say that. Let's say attached earlobes, okay? And so there's a gene for attached earlobes on mom's chromosome. There's a gene for attached earlobes on dad's chromosome. Looks like if this was my chromosomes, like this is what my somatic cells, my body cells had in them, it looks like I have the same allele for both parents. That is called homozygous. And it's homozygous recessive because they're both the little r, okay? So that's what that means. This is homozygous dominant. And then, ooh, get out of the way, little toolbox. And then down at the bottom, there's a heterozygous, if it would calm down and make that go away. So down at the bottom, there's a heterozygous one that you can see the maternal line has the dominant, and the paternal chromosome has a recessive. And so let's pretend like that cystic fibrosis. Cystic fibrosis is a disorder where a protein that is in the channel in your cell membrane, the channel protein, is not getting rid of salts, certain salts. And they're building up. You get a mucus building up. It's really pretty horrible disease. You get mucus buildup in your chest and your bile ducts and in your intestines. You just can't breathe. It's pretty horrible. So in this case, if this was cystic fibrosis, then the paternal chromosome, the purple one at the very bottom, you see a little recessive C, the little baby C. That would be a mutated DNA, so mutated protein, non-functional, not working. That person would be sick, but the maternal chromosome has a capital C, which makes the correct protein. So those are different alleles for the same gene. And so since the mother's makes the actual good working protein, you don't notice it. Like that one works good enough that you are not sick at all. Like you're what we call a carrier. You're carrying this recessive, this bad, quote unquote, it kind of is bad because cystic fibrosis. You're carrying that, but it's not expressed in you. Like you're not seeing that trait. You're not seeing that phenotype, that physical expression, because the maternal chromosome had the working protein and it works good enough. And so that's what it means whenever we say dominant. Dominant means if you have one, so like in this case, cystic fibrosis is recessive because you have to have two of them in order to show the phenotype. If you have one of the normal protein, then you don't have cystic fibrosis. Okay, so the normal is dominant and cystic fibrosis is recessive. Okay, but there are some genes, some like the hanging downy earlobes. I don't have earlobes; they're really stuck to my head. We've already talked about this. Why do we keep talking about my earlobes? Anyway, so basically, the deal with earlobes is that if I had one gene that was like the dominant, like if I had one gene for the free earlobes, they call them, then I would have had free earlobes. But I didn't have one gene. So the dominant would be free, and I only take one to make that developmentally happen in my ears. All right, let's see. I think that's it. There's a couple of questions here. You can pause the video and see if you get the right answer for that one. By the way, it's supposed to be a heterozygous green pea. I forgot to write that on there. So that should be a heterozygous green pea. So now pause the video. Okay, now you should be done, and you should have found that that you had 50% chance. Okay, what would be your genotype ratio? Pause the video. Okay, you should have found that it was 0 to 2 to 2 for genotype ratio. No homozygous dominant, 2 heterozygous, 2 homozygous recessive. Okay, all right. And what's your phenotype ratio? Pause the video. Okay, you should have found that your phenotype ratio would have been 2 to 2. There's no 0 at the front because there's only 2 options. Green is dominant, yellow is recessive. You have 2 that are green, 2 that are yellow. Okay, here's another one. Go ahead and do this one. Pause the video. Okay, you should have found that they were 100% gray, all gray. And what would be then your phenotype ratio? You can pause the video if you want. Should have been 2 to 2 to 0. 2 homozygous dominant, 2 heterozygous, 0 homozygous recessive. And then genotype ratio, 4 to 0. All right, let me know if you have any questions. I'll see you soon. Take care of yourselves. Bye-bye.